depending upon where we are, the way they're arranged. There's what's called single unit smooth muscle, and if you notice here, is that there are lots of smooth muscles cells that are connected by gap junctions. So these are physical connections between the two of them. So the reason we call it single unit is because all of those cells function as a single unit because they're all connected. Right? So I can't regulate it at the level of an individual cell. And whether you have single unit or like this, multi-unit, here notice that none of the cells are connected. We call them multi-unit. How you regulate that just depends on where you are in the body. Some things need to have the multi-unit regulation like this for finer control, like arterioles and things like that. Others have single unit, like circular smooth muscle in the intestine or something like that, to allow for peristalsis. It really just depends on what level of regulation we need. Not a big deal, right? The point is that it's still going to respond to neurotransmitters. Now, this is the autonomic nervous system here controlling it, right? So this would likely be sympathetic, right? Branch of the autonomic nervous system to trigger contraction, unless we're talking about the digestive system, right? In which case this would be the parasympathetic to stimulate contraction and motility there. Now, that just shows you the duration of the time it takes for a contraction in smooth muscle. We mentioned that skeletal muscle over there can contract in the neighborhood of 250 milliseconds or so. Right? You can see there, the one in red there, it's about a quarter of a second it takes. Smooth muscle takes nearly 5 seconds for a twist. So contract, relax. So it's very slow. And part of the reason it's slow is because we got to have time for this cascade to occur right here. Okay? We know those things take time. Okay? All right. Still has actin and myosin interacting with each other. No big deal there. You can see the arrangements of them here. No sarcomeres, but you still have to have areas where myosin and actin overlap so that they can pull together. It's kind of interesting. It forms this mesh inside the cell, this kind of network, if you will, so that when the muscle contracts, the whole thing kind of just bunches up and squeezes together. So it doesn't shorten really in a single plane like we get for myofibrils and things like that. All right, here's the cascade. Now let's focus on this. This is just following A happens, then B happens, and whatever. We know that this starts with an action potential on the sarcolemma. So we start with an action potential on the sarcolemma. Okay. What we said was different was that there was some calcium influx from the ECF. So on the sarcolemma of smooth muscle, there are voltage-gated calcium channels. And these are just like the voltage-gated calcium channels that are on the axon terminals, right? They are closed at -70, although I think that resting potential in a smooth muscle cell is more like about -60, but it doesn't matter. But they're closed at resting potential, and then they open with an action potential. So if this cell is getting a signal to contract, the voltage-gated calcium channel opens. And you get some extracellular calcium coming in. Now, if we didn't open this channel, or if this channel couldn't open, you don't get much calcium coming in, you're not going to get nearly as much muscle contraction. Eventually, the depolarization of the membrane would trigger depolarization of the SR, but what really triggers depolarization and the opening of calcium channels on the SR is this calcium influx. Right? So look what happens. Action potential here, calcium from ECF. As soon as the calcium from the ECF comes in, depolarizes the SR, and then opens that channel. And now you get a whole bunch of calcium coming out there. Okay. In terms of amount of calcium for contraction, maybe 20% or so of it comes from the ECF, and the rest is from the SR. Okay. But the point is that you get this extracellular calcium supplement allowing for this. Right? And as soon as you get calcium present in high enough amounts in the sarcoplasm, we can trigger that calmodulin MLCK cascade. And look what happens, it's a piece of cake. Calcium activates calmodulin. So see, calcium binds to calmodulin and it's activated. As soon as calmodulin is activated, myosin light chain kinase is activated. As soon as myosin light chain kinase is activated, it takes phosphates and puts them on myosin to activate myosin. As soon as myosin's activated, you get cross bridges and power strokes. So in a relaxed muscle, what do you have? In a relaxed muscle, you have inactive myosin, no cross bridges. You have inactive MLCK. You have inactive calmodulin, and you have no calcium. So calcium still triggers muscle contraction, right? It is a rise in cytosolic calcium that triggers contraction. It just does it differently. Okay? So we get a rise in cytoplasmic calcium. Calcium binds to calmodulin. Calmodulin now activates MLCK. MLCK phosphorylates myosin. Myosin binds to actin and a power stroke. What do you have to do to relax this? Well, we need to remove the calcium, right? Pump the calcium back into the SR, pump the calcium back out into the ECA. Now you have what? Inactive calmodulin, and you have inactive MLCK. Now, interestingly enough, inactive MLCK, guess what its other name is? Myosin phosphatase. So what's it going to do?
Remove.
Take the phosphates off of here, inactivate myosin. So the same enzyme controls it, but it's a kinase when it's active with calmodulin. Inactive form here is a phosphatase. So what's it going to do? It's going to remove all the phosphates, and will the muscle relax or contract?